welcome to today's ECHA Science Seminar. That's Wim de Koen and Jan Weber. We are very happy to present you today Professor Thomas Backhaus and Dr. Romana Hornek-Gausterer talking about addressing chemical pollution in biodiversity research and management. So, what are ECHA Science Seminars? It's a series of online events with scientific content that's relevant to our regulatory activities and will cover both human health and environmental topics. And here we aim to discuss science, its regulatory relevance, and how we can possibly uptake the results within our mandate. So today we're talking about chemical pollution, which is known as one of the key drivers of a decrease in biodiversity. And to reverse the degradation of ecosystems, the Commission adopted its new biodiversity strategy for 2030. Still, it's unclear if those RRM, those risk management, regulatory risk management procedures, if they're able to predict, mitigate, and prevent the adverse effects on biodiversity, or how they can do that. Also, we need to better understand how we can embed those new approach methods into current risk assessment as it's done under the, regulatory frameworks. And that's why we invited two key experts on the subject, namely Professor Thomas Backhaus and Dr. Romana Hornek-Gausterer. And Professor Backhaus leads the Institute for Environmental Research at RWTH Aachen University, and he's the chair for the Ecotox and Environmental Risk Assessment department there. He has a passion for toxicology and ecotoxicology of mixtures, and we are going to hear from him very soon. Dr. Romana Hornek-Gausterer is Program Director for Sustainable Environmental and Bioprocess Engineering at Vienna University for Applied Science. She works on a PARC project on the environmental safety assessment under the Commission's roadmap towards replacing animal testing with new approach methods. She's very interested in translating science into policy, and I'm sure she's going to show that very soon in a few minutes. So now I would like to hand over the floor to Romana to start the lecture. Thank you.
Thank you. I'm very happy to have today the opportunity to give a presentation together with Thomas Backhaus on this topic. So we directly dive into the topic. So the question is actually, what is biodiversity? And I will try to explain it. So biodiversity is the variety of natural life and includes all living organisms found in land and in water. Important is that all species have a specific role and we depend on these organisms, from really tiny microbes to really larger animals and plants. Biodiversity includes genetic diversity, so species diversity and habitat and ecosystem diversity. And it's important that genes are really the building blocks of life. So they determine the characteristics of all living organisms. And maintaining genetic biodiversity by conserving species and all the varieties is really a cornerstone of nature conservation. So conservation of genetic biodiversity is central, but there is also species diversity, so the number of species in different ecosystems, and there is also habitat and ecosystem diversity. So the variety of habitats, such for example, forests, oceans, deserts, and wetlands. So why should we actually care about biodiversity? Our society depends on healthy biodiversity. It has a really profound impact on human well-being in many ways. So it delivers key ecosystem functions. And it underpins also the ecosystem services we all benefit from, like clean water, food production, but also pollination. And it has also an economic value. And for example, the World Economic Forum recently published an edition of a global risk report in 2025, and it analyzed the global risk through various frameworks. And biodiversity loss and ecosystem collapse rank as the second most severe risk, highlighting, first of all, the importance, but also there is a value stating behind. So, and this value is really enormous. And when we go into the details, what is actually the main issue on biodiversity loss and how much does it happen? So what we see is there is a dramatic decline, and the decline is 73% in the average size of monitored wildlife population just over the last 50 years. And freshwater populations are the most affected, and they decline by an average of 85%. So, but the decline, and as you can see it here in this slide, is very different, and it depends on the region where you are, and the range is between 35 and 95%. The steepest decline in monitored wildlife populations are recorded in Latin America and the Caribbean, followed by Africa and Asia Pacific. So we as humans, we have, of course, we are also drivers for the biodiversity loss, and the anthropogenic drivers. These are those which are the result of human activities and decisions. They include, for example, land use, climate change. But one important driver is the pollution. The pollution, which has a negative impact on our nature. And you can see there is a difference, the difference on impacts across various taxa, but also across various regions. The European Commission is, of course, intensifying the effort to protect and restore biodiversity, as highlighted in a series of strategic documents. And there is the EU Biodiversity Strategy 2030, which was launched 2020, which aims to put Europe's biodiversity on the path to recovery by 2030. And but what we have is, for example, when I look at different regulations like REACH, biocides, pesticides, or CLP, all aim to prevent really adverse effects on the environment and ensure a high level of protection. But despite the Commission efforts and the regulatory frameworks, they do not often explicitly define biodiversity, and also not how to assess really the impact of chemicals on biodiversity. For example, for REACH and CLP regulation, they do not refer to biodiversity or biodiversity-related concepts of diversity, including the ecosystem, as a protection goal. This is only the case for biocides and for the pesticides regulation legislations. To capture really what is ongoing in Europe, there are some key objectives, of course, of biodiversity conservation. And there were some indicators established, like, for example, the Common Bird Index to keep if we can reverse the decline of birds. And there are some other indicators, for example, terrestrial and marine protected areas and the EU forest connectivity. So these indicators have been provided, and the goal is to reverse, so to say, the impact by 2030. But what we can see is, in summary, all the indicators I just have presented, Europe is not on the track to meet any of the 4 biodiversity and ecosystem related monitoring targets. And what is, I think, very important that, you know, we have, of course, these essential biodiversity variables. There are a lot of variables where you can measure ecosystem structure, function, community composition, and so on. But this is not linked with the legislations we have in place, for example, REACH or CLP. So when I come to the challenges for environmental risk assessment, I would like to highlight that we have some silos, and the silos are occurring in policies, but also in regulations, but also in science. And I would first like to go a little bit through environmental risk assessment and why it is not on track. There are fragmented data. So we have the biocides regulation, we have REACH, we have the pesticides regulation, so substance-oriented regulations. And the data are really there, but they are fragmented, they are not connected, and regulators also are not connected. So it is also the assessments. And we have a lot of blind spots, and I would like to mention some of those. We have indirect effects, but we have not only biocides or pesticides or industrial chemicals in a river, we have a lot of chemicals and also the transformation products in a river, in a soil. And we are not assessing them together. We have long-term effects and effects based on very low doses. And of course, sometimes we have out of scenario exposure. And the problem is we have an impact on all the total use of chemicals, meaning industrial chemicals, biocides, pesticides, and so on. And when we look at the regulations, we have in place hazard assessment, exposure assessment, we are deriving PEC values, but actually there is no cross-check of all the methods we have and the outcomes with real field data. So do we validate what we have assessed during regulatory assessment? And actually at the moment not. And then one example is one substance, one assessment. I think we are far away from that. And I will give you later on an example. For many substances, we have lack of experimental data. We do not have data on the exposure. We do not have data on the hazard. And we have an increasing volume, meaning we have an increasing volume of chemicals which are on the market, biocides, pharmaceuticals, and so on. So the pressure on the environment is steadily increasing. And of course, we are assessing one substance by the other. We are also grouping substances, but also problematic is normally we extrapolate, and this is done within REACH, but also for the biocides. We extrapolate from very few species to effects on population and the entire ecosystem. And we do not take into account many other stressors. And this consideration is really urgent needed. Very often we focus on the aquatic compartment, and there are gaps in the terrestrial compartment and also on the ecosystem level interaction. We have some organisms which are missing, model organisms, but also models for these organisms to go into the direction of new approach methods. And we have, of course, effects on organisms which were not intended, so effects on non-target organisms. And there is also one important effect also for biodiversity is behavioral effects, so the integration of behavioral effects. And enforcement and management measures are at the moment limited. If we do not control what is ongoing in the environment, and I think this is also important, the risk assessment is getting even more complex. We have more, or there is more workload, so lengthy evaluations, delayed decisions, and also delayed implementation. And even if we are measuring in the environment some concentrations of chemicals, either in compartments or biota, there is no feedback possibility to upstream regulations. And there is one project ongoing within PARC, and the question was more or less, is what is done under, for example, the biocides or under chemicals legislation really good? And does it protect the biodiversity? It is questioned by regulators. And the same is also true if we look on scientific challenges. We have not included ecological perspectives. So each discipline does some scientific reports and research, but it's seldomly interconnected. So it's very important that we understand the effects, but also the effects on the organisms, the effects with all the other stresses, and on ecosystem levels. So we need really joint forces. And the next here, I think it's very good seen. It's a recent report from the Joint Research Center from the European Commission. It's protection of biodiversity as the ultimate goal of environmental safety assessment, and how does chemical pollution affect biodiversity? And I think this is really, really quite good here. Under substance-specific regulations, we are performing a lot of ecotoxicological measurements. But the problem is we need to link these laboratory tests really with what's happening outdoors. We need to include ecological factors. And then I will give you some examples. So the first example is we are using species for risk assessment, but we are using only a few species. So mostly fish, daphnids, and algae, but we have many species, but we have also interaction between the species, and we have several abiotic factors which affect the outcome. And of course, we have multi-chemical exposure, and not only one ecosystem, but many ecosystems. This is one example, the first one. The next example I would like to highlight is following. There was a study within PARC, one substance, and we have different regulations. So like, for example, the pesticide regulation, the biocide regulation, and also REACH. So the effect assessment is really similar within those regulations, but the outcome was not the same. So we derive with one substance 5 different threshold values. So, and the amount of effect data within these regulations varied significantly. So here also one substance, one assessment, and to have all the data together for the substances is, I think, really, really necessary. One other example is the exposure. We have many models for exposure assessment, but the question is, have they ever been verified? And are they really, really working? And at the moment, so to say, simpler models are even perhaps better, and there will be a report coming out in May. And then this is the final slide. Afterwards, I hand over to Thomas. I would like to mention there are a lot of things ongoing. So there is ECHA's key areas of regulatory challenge. And there is also the biodiversity mentioned, where we need methods like new approach methods and to bridge the gap between science and regulation. But the same is also true for EFSA. So also NUMS should be integrated. And there were several workshops workshops ongoing. So how could we, with the methods we have or new methods which are coming, better protect the environment and the biodiversity? And with that, I hand over to Thomas.
Okay, thanks and good afternoon, everybody. Thanks for giving me the opportunity to talk a bit about biodiversity effects here. The problem with those seminars, as much fun as they are, is always that you don't really know who's in the audience. So please forgive me if I'm preaching a bit to the choir. If that's the case, my apologies, grab a cup of coffee, but maybe there are at least one or two examples in here that are sort of interesting. So I'm going to walk you through a couple of examples, which I think are highlighting key issues on why we are struggling to protect biodiversity from chemical pollution. And then at the end, I'm going to summarize that. And then I'm switching to another set of slides talking a bit about the Water Framework Directive as one example of the recipient-oriented regulations. Okay, given that I'm assuming that I'm basically talking to people that have been working in chemical risk assessment for most of their lives, I'm not going to start the talk with talking about DDT and the examples and the experiences that we have been collecting here. That's just a reminder. That's a photo from the 1950s where basically school children were treated with DDT while having lunch in order to treat ectoparasites, which is also sort of lowering biodiversity on those humans. But this basically is just a reminder that even those decisions that we had at that time, we are still struggling with the consequences even now, 80, 90 years later. But that guy wants to have a talk with us. That's an Indian vulture, and that is basically the say most severe impacts that we've ever seen from chemical pollution on vertebrate species ever. It's far exceeding any problems that we ever had with DDT. And the main surprise there is actually the chemical that was driving that. And the chemical is diclofenac, which you might use in painkillers. Antrotec, Voltaren are typical brand names. There are almost 100 different products on the market. So these are just the most common names. It's a typical painkiller that is used to treat especially arthritis and rheuma. So long-term treatment, but also the typical hangover and stuff like that, but mainly joint pain. The thing is, the risk assessment that we're basing everything on is based on the idea that that compound is exclusively used as a human medication. But because the compound is out of patent, it has been also used extensively in veterinary medicine in Asia and especially in India. And the problem is that the compound, one of the side effects is kidney toxicity. And it turned out that vultures are extremely sensitive to that side effect. And they were literally dropping dead out of the sky because they were just dying from kidney failure. And what nobody saw coming at that time is simply that there is actually an exposure pathway from a human medicine, from a human pharmaceutical to into the ecosystem to vultures, because that seems to be a very remote chance that that ever happens, and it actually did happen. And the reason for that is, of course, that because we can, the reason there is actually that the cows and cattle in India have a special role in society there, and that basically means they are treated for exactly the same reasons as human beings with that particular pharmaceutical. And what you can see here on the right-hand side is, I don't know whether you can see my pointer. I guess not, at least, there it is. I hope you can see my pointer now. What you can see here is the population index of those vultures. And what you can see is that over the years, how drastically that has been declining. That's a log scale here. So we have been basically, reducing the population by 99%. And whether we, now that we know what's going on there, actually see a recovery of the population, that depends a bit on how optimistic you are actually modeling the data. You could also basically say nothing is happening over years now. The compound has been banned more or less for veterinary pharmaceuticals in India also around 2007, 2008, and yeah, we see that not a lot of things are happening here. So they are on the brink of extinction, which is one major problem, of course. But what we are also seeing are huge impacts on human health. Here we are really seeing the connection between the impacts on ecosystem level and the impacts on human health. The problem is that vultures are playing a critical role as scavengers in those ecosystems. And if that scavenger is gone, because they are just dying out, the role of scavengers is taken over by feral dogs and rats. And that increased dramatically the rate of rabies, but also of water pollution by carcasses lying in water supply areas. Also, it has sociological consequences. The so-called sky burials of the parsi population obviously also don't work anymore. There was just a recent publication in 2024 where they have been really trying to disentangle how that works. I'm not going into the details there. The reference is there if you want to read up on that. I just want to point out the quantity, the consequences that we're seeing here. What we're seeing here as a consequences of that impact is that we have an increase in human mortality by over 4%. And that is really not a little. That's a lot of people that were just dying because of increased water pollution, because of increased infection by drinking polluted water, by increased rates of rabies that largely go untreated. The economic damages are quantified at 70 billion per year. So I think even if we put aside the ethical considerations of protecting biodiversity, simply from a human health perspective, that is really, really critical that we actually keep that in mind, that we are ultimately dependent on on a functioning ecosystem. The problem is it took 6, 7, 8 years to find out why are those vultures dying, because nobody first suspected, oh, it's a pharmaceutical, it's a painkiller, it's something that you take against joint pains. So why the hell does that? And so the environmental forensics behind that take a lot of time. And there's another example. So now we're switching from the southern part of the world up to the say more northern hemisphere. So here we're looking at the western part of Canada. So we have Vancouver here, and here we have the Puget Sound, which is a small bay, or it's a very narrow bay, and a lot of salmons are migrating upwards and then into the different rivers here. And what people have been discovering there is that those salmon, as soon as they enter certain rivers, not all of them, but certain rivers, they also simply dropped dead. They simply died. So here's a description of what was going on there. The daily surveys in those streamed indicated that we have mortality rates that range from 60 to 100%. So between half and all the salmons entering those streams were dropping dead. Comparable rates usually are below 1%. So there was a very clear signal something is really going on there. And if you find that, you start to explore why is that the case. So conventional water quality criteria, pH value, oxygen saturation, etc., turned out nothing. Pesticide exposure was not a problem. There were no diseases found, and the condition of the animals looked okay. So they were entering the rivers in a healthy condition, and then they died. So it wasn't any disease that they brought with them. They also looked at exposure to metals and at petroleum hydrocarbons, because they realized it's mainly in urban streams where that is happening, and all of those things actually didn't turn out anything. So the conclusion was there's something that is as yet unidentified as a toxic contaminant here. And the question then really is, what kind of compound is that? And it took a lot of time and effort to find that out. They implemented what is called a toxicity identification and evaluation strategy, or EDA strategy, where you start with a full leachate, and then you are basically actually going down and fractionate that in order to find the compound that is actually driving the effect there. Could I just get a second? I just need to take care of something. I'll be back in 2 seconds.
So while Thomas is back in a sec. I just would like to remind you, please use the chat anytime to drop questions or comments. And it's going to be like 60 minutes in total of presentations around, and then we have hopefully 30 minutes for discussion. Thank you.
Yep.
Thomas,
Thank
back.
you. I'm back. Thanks for that, and sorry for that. Okay, so that was a lot of effort to actually try to find out which compound that is, to start with a full leachate of the stuff that you are identifying in that river system here, and then you fractionate that according to lipophilicity, according to ionization, according to molecular size, and all of that. And then in the end, you are hopefully identifying a fraction and a compound that is driving the toxicity to those salmons, to those fish. That's an extensive, quite labor-intensive approach where you also need to run a lot of animal experimentations. But they managed to identify the culprit in the end, and that is a degradation product from 6-PBD. Here is the confirmation experiment, the dose-response curve to salmon in a lab context where you just expose them to that particular compound. And then here you actually have the EC50 value for the runoff and the leachates from the different sites in those rivers that were affected, readjusted to the concentration of just this particular compound. And you can see the LC50 under lab conditions is 0.8 microgram per liter. And you see that they are the complex mixture there is just driven by that particular one compound. So it's 0.8 microgram per liter here also. So that is a real confirmation. So the question then is, of course, where does that compound come from? So what is that for a chemical? And that goes together in the end quite nicely. It's a compound that is used in car tires. It's an antioxidant in car tires. Here you see the reaction pathway. It's scavenging ozone in order to protect the rubber and the polymers in the tire and to prolong the lifespan of car tires. And that is exactly what you're seeing here. And it's not the parent compound that is particularly toxic, but it's the degradation product after the compound has been doing its job. It's used between 0.2 and 4% in car tires, which is a substantial concentration. And given that we have a lot of cars running in those urban streets, and then we have a lot of runoff in the urban streams, that's the exposure pathway, and that's the compound that is basically then killing those animals. And what you can see here is really how much more toxic the degradation product is from the original product, from the original compound. So here's the parent compound. We have an EC50 for this particular salmon species that they have been testing there. And the EC50 drops from 250 to 0.8. So that's a difference of, I don't know, 300 or so, factor of 300 or something like that. So this is really a massive increase in toxicity. So that basically goes to show how the complexity of exposures are really posing a challenge because it's sometimes almost impossible to keep all the use patterns, all the use scenarios that a basic chemical is, the scenarios in which the chemical is used to actually have that in mind when you're doing the risk assessment. That's a huge challenge. There's another challenge, and I'm just going to show you one example there, and that is that the environment is actually basically opening up for the possibility of indirect effects, and that is sometimes coming around to bite us in our behinds. And here's the reason that I just, the example that I just wanted to show you. We have a pathogenic fungus that is one of the main causes for the worldwide decline in amphibians. It's basically a deadly disease for those, for all the different amphibians, and they are just dying on that. So what people have been starting to do is they use a standard fungicide, a standard triazole fungicide in order to clear the fungal infections and in order to basically give them a better chance of survival. So what they have been doing is basically, let's hear an example from the National Museum of Natural Science in Madrid. They have been capturing the tadpoles and then basically exposing them under lab conditions with the fungicide. in order to get rid of the infections. At the same time, the ponds in which the tadpoles would be living is drying up and then refilling by rainwater so that the fungal infections should be gone. So the idea is to disinfect the organisms, to let the ecosystem undergo a natural kind of sterilization, and then you should have a higher survival rate of the fungi. The problem is when they did that the next year, all the treated animals had become infected again. So it just didn't work. And the question, of course, is why didn't that work? We know the tadpoles were basically infection-free when they have been put back into the pond. So what's going on there? And you can see that very clearly when you follow that up with lab-based studies. So here we have the treatment of the amphibians. Here you have the abundance of infectious colonies that you find on the skin. You have the control animals. You have a bit of a variability on how much they are affected. And when you treat them with fungi, you have at least a factor of 10 lower infection rates. Again, it's a log scale here. So that's a substantial success of treating those animals. So that's exactly how that was planned. But when you then wait a while, what happens even under laboratory conditions is this. So what you see is that the control, we actually have a decreasing level of infections here, and that is simply because they are basically developing a natural immunity. But when you treat them with fungicides, after a while, you have a drastically, and we're talking orders of magnitude, higher level of infections here than you had before. So you treat them with fungicides and you end up with 100,000 times higher infection rate. So massive effect here. And the reason for that is it turns out that the fungicides are not only killing the fungi, they are also toxic to the immune system of those frogs. So you make them actually far more vulnerable to the few spores that are surviving the treatment. And then basically they are very vulnerable, and then you see those enormous increase in infection rates. So you can also look at how things are happening in the field. You can just correlate the fungicide concentrations in either frog tissue itself or just in the wetland in which those frogs are living. And you correlate that with the infection prevalence, and you see a clear correlation. Of course, there is a huge uncertainty behind that, because that's a variability of exposures and of animals that you capture in the wild, but you see a very clear statistically significant correlation between the amount of fungicides that they are exposed to, and that is agricultural fungicides in the wetlands where there's also farming nearby, and the infection prevalences. So although we are having a fungal disease here, fungicides are playing a critical role, because those fungicides are lowering the immune system, the immune status of those exposed animals, and that increases the vulnerability of the population to a sort of natural non-chemical stressor. So those indirect effects are a huge challenge when you want to basically describe what are safe conditions for use for those chemicals. Okay, I can go on with a lot of different examples of why and how we are seeing impacts in the wild, but I'll try to just summarize a couple of key points, I think. So the one thing is we also should remember that a lot of those chemicals are actually used for reducing biodiversity. We have pesticides, we have biocides, we have pharmaceuticals that are all meant to kill certain organisms. And that is definitely something that works. We want that effect when we are trying to kill infections. But we also should remember that the whole idea of agriculture is to actually lower biodiversity. We don't want to have a high, above ground at least, a high biodiversity on our agricultural field. We want to give our crops the advantage there, and that's the species that we want to promote, and we want to kill off the others. So the whole idea here is to actually reduce biodiversity, and that works. The problem is it works perhaps a bit better than we intended. We have an enormous similarity between the target and the non-target organisms. That's always posing us a challenge. We spray insecticides in order to kill insects In an agricultural setting, we want to kill those insects that are feeding on our plants, for example, but at the same time, we like bees. But of course, bees and those other insects, they are basically the same thing. So it's really hard to differentiate that and to come up with treatment schemes that acknowledge that. We cannot test all organisms. Nobody thought of testing the toxicity of diclofenac to Indian vultures, but that is turning out to be the problem in the end. So there are always organisms that are particularly surprisingly sensitive. We don't test all the chemicals, especially not when it's metabolites, degradation products, and stuff like that. So there are largely untested chemicals out there that are unexpectedly toxic. We do not account for all the exposure pathways, the human pharmaceutical to the cattle, to the vulture. That is definitely something that nobody had on their radar. Exposures differ drastically in space and time. Obviously, we have those indirect effects where I just gave you the example of the fungicides. They are extremely hard to predict, and they are driven by the specific nature of the local ecosystem where the exposure actually takes place. And then we have a lot of management issues, also lack of enforcement, regulatory gaps. We just talked about that. That's something that we also need to discuss. So I think we have a lot of examples where intended exposures impact biodiversity as intended. So that's a good thing, but we need to learn from that. We need to see how that actually plays out and why that happens and how we make use of that. The best studied examples of the consequences of unintended exposures are still increasing carbon dioxide level from ocean acidification to climate change, but also eutrophication. That's the two examples where we have large-scale, enormous consequences. The other area is where individual species are impacted by particular point sources and by chemicals with a known high ecotoxicity, pharmaceuticals, pesticides, biocides, compounds that are made to be biologically active. Our big knowledge gap is all the other types of chemicals that are used everywhere all the time that are never intended to be biologically active, but just by chance, because we're using so many different chemicals, some of them are also biologically active, like the degradation product of an antioxidant used in a car tire. That plant, nobody intended that, but it still was biologically highly active. And that's definitely a challenge to account for that in our risk assessment. Then I'll just continue with a more regulatory perspective on things. So what we have, and again, now I'm really preaching to the choir, we have three different types of regulations that we are concerned with. We have the substance-oriented regulations, and the main ones that we are usually talking about is REACH, pesticide, the biocide regulations, and the pharmaceutical regulations. Cosmetics should also perhaps be mentioned here. But then we have the recipient-oriented regulations, and the two main directives that we use here is the Water Framework Directive and the Marine Strategy Framework Directive. So the idea here is not to look at individual chemicals and to identify conditions for safe use, but to protect a particular ecosystem that is the recipient of all those chemicals and other stresses on top of that. Then we also have process-oriented regulations like the Industrial Emissions Directive, and I'm not going to talk about that for the sake of time. The concerns for biodiversity when it comes to all of that is actually not something new. We have a paper talking about the sinking arc pollution and the worldwide loss of biodiversity from 1992. And we started to basically talk about the impacts of chemical pollution on biodiversity and ecosystem services. One was that 10 years ago or something like that, together with people from AstraZeneca and from the US EPA, where we basically said, okay, folks, we really need to understand better what's going on without understanding who's there, what's their ecosystem function, and what are the affected ecosystem services. We can't really start doing assessment strategies that is going to protect that. In Europe, the Water Framework Directive is one of the main regulatory frameworks when it comes to the water environment. I just would like to stress that that we have a very, very close connection also to the protection of human health here, because water is the source of our drinking water, and we also have indirect exposure via eating fish and other seafood. So I'm going to switch over that, or I'm going to skip over that for now. The assessment of water bodies is usually done on two levels. We have the so-called chemical status, which is basically an assessment whether so-called priority substances are above or below the EQS value, which is the environmental quality standard, which is loosely the same as a PNEC. There are fundamental differences, but the underlying philosophy is the same thing. It's a concentration that is supposed to not impact any aquatic life at all. And then we have the ecological status, which is basically looking especially at physical-chemical quality elements like nutrient content, for example, but also acidification and things like that. But especially we have the biological quality elements, and that is asking the question whether at a certain stretch of river, a lake, or a coastal area, we have those organisms being present that are supposed to be present under good conditions. And then you basically look at that, you put that all together. I'm not going into the details on how this is done. It's basically that the lowest criterion is driving the assessment. And then you end up with a scheme where you start to classify the ecological conditions, something from high to bad status. And you do the same thing for the chemicals, where you have a good or a bad chemical status. And if we're just looking at how the status is of aquatic bodies, aquatic ecosystems in Europe, and that's the data from May 2024, that's the newest one that are published by the Environmental Agency in Copenhagen. What we can see here is the chemical status, and we see that around 10% of them are actually having problems to achieve a good chemical status, which means that in around 10% of the water bodies in Europe, we know that chemical pollution is at an unacceptable level. We have around 2/3 where we know that the chemical status is actually good, and we have still a substantial gray area, especially in transitional waters here. When we're looking at the ecological status, so how good is the ecological situation, we also see that we have a poor or bad in around between 10 and almost 20% of the different water types. We have a huge chunk where the status is good or even excellent. And we have some waters where we still need to do more biological monitoring. But what we see, excuse me, what we're seeing is that in around between 10 and 20% of the water bodies, we have a problem, and we need to think about what we need to do there. And the question, of course, before you start thinking about what to do, is the question, who's causing those problems? And here's one example where people have been looking at the status of rivers in Europe, and they've been trying to tease out, using all the monitoring data that are produced by the Water Framework Directive, which are the causes for the impact that we're seeing. So we have land use patterns that have an impact. We have the water flow that obviously has an impact on the ecological situation. Here we have eutrophication, total nitrogen, total phosphorus in green, and then we have the toxic substances. And what we see is that they are accounting for around 25%, so 1/4 of the impacts that we are seeing on those river types. So they're not the only stressor that aquatic ecosystems are experiencing, obviously not, but they are one of the main players that are really driving it. And if you zoom in a bit more to that, then what we can see here is a correlation analysis. We have on the x-axis basically the concentration of the chemicals, the overall concentration of the toxic chemicals in a water body, adjusted for their ecotoxicological potency. So basically 1.0 would be a concentration where you expect all the creatures in a river to be severely affected, and 0 is obviously no pollution. So that's not a concentration in microgram per liter or something like that, but that's the so-called MSPAF, which stands for multi-species potentially affected fraction of species, which is basically you take laboratory data, you take algae Daphne fish data and whatever you find for the different compounds, and you translate that into a toxic pressure on an ecosystem. And here you see the ecological status based on the monitoring data under the Water Framework Directive, and then split up according to different river types. If we just look at the overall picture, and what we can clearly see is that the average condition of a river is really dependent on the compounds that you have here. Or in other words, if you have a river in an ecologically bad situation, then you actually also have a high concentration of toxic chemicals. It doesn't mean that's the only stressor, I mentioned that before, but there seems to be a clear correlation that is driving it. And one particular challenge here is that we are not looking at just one chemical and we correlate that and we do environmental forensics and then we find the chemical and then we regulate the chemicals and then we're done. It's the mixtures. We know that for a long, long time that chemical mixtures, the effects, the toxicity of individual chemicals is actually adding up to a mixture risk that is substantially higher than what you expect from the individual chemicals. And the Joint Research Center actually has done that already 10 years ago or something like that, where they have been looking at our environmental quality standards. Are they really safe? Are they really protective if we're dumping those chemicals together the same way they are occurring in any major river? And here's just one example, and I'm just showing you this one example where we have two algal species, so one of the classical biological quality elements, at a concentration that equals exactly the sum of all the EQS values. So every concentration is at its absolutely safe level. For the sensitive species, for one of the green algal species, we already have a 20% impact on primary production, on the photosynthetic activity, which is basically providing all the food in an aquatic ecosystem. Slightly less sensitive species, they start to be affected only at higher concentrations, but even those, if we're just increasing the concentration to tenfold the EQS concentrations, then we have a 40%, and for the most sensitive, we have more than 60% effects here. So that's a clear indication that even those few priority substances, that's just at that time it was 30-something, 35 or something chemicals, if we mix them together at safe concentrations, we are ending up in a situation where we can expect ecosystem-wide consequences. And we're seeing that with those correlation studies. And of course, we have the problem that those compounds are coming from very different regulatory areas here. This is more looking at things from a human perspective, but all those processes, all those groups of chemicals are also environmentally relevant, and all of them are actually adding up in that recipient, in that particular ecosystem, which really leads to consequences. And we need to better link those regulatory silos together in order to do something about that. And we're facing a couple of particular problems. So the one particular problem is the mixture exceeds the risk of the individual compounds. That has been proven over and over in I don't know how many dozens of empirical studies. But we also have another problem. And here's a study that we did with chemical monitoring data for pesticides in Sweden. It doesn't matter what kind of compounds those are. It's just for pesticide, the monitoring efforts are most advanced. And I don't want to go into all the details here. These are 6 different sites in the southern part of Sweden where most of the agriculture takes place. And what we have been doing here is a mixture risk assessment. We're using the same risk quotient or hazard quotient, as some people call that approach, as we do that for individual chemicals. So if the PEG-PNEC ratio, to talk about in breach terms here, is above 1, then we can't guarantee safe use. And if it's below 1, then things are okay. And what we can see is that in a lot of those sites, we have concentrations that are above 1. That's not very surprising that in rivers that run through agricultural landscapes, we have a risk. What I want to point out here, what the critical issue here is that if you look at, for example, this area here, it's basically constant over decades. We stopped analyzing that in 2013, and I can promise you it continues exactly the same way. It's just boring to replot that every time. And at the same time, especially in the years 2008, 2009, 2010, we had a massive restructuring of the pesticide market. We have lost a lot of compounds that have been taken off the market for environmental concerns. And the only thing that happens is that they are replaced by other products that are used at concentrations that end up adding the same toxic pressure to the environment. So our phasing out of individual chemicals doesn't really help us to move the needle, because they are just replaced by other compounds. And the farmer doesn't really care what's in the product that he's spraying on the field. And the environmental organisms don't really care that now it's a new compound that is doing the same thing as the old compound before. So this substance by substance phasing out, banning them, etc., is not really helping us to improve things. Things are flat, constant all the time. The Water Framework Directive is under revision.
Thomas, I'm sorry to interrupt you. It's just we have roughly 5 minutes left of the actual lecture. So if you could together maybe wrap up within the next 10 minutes, that would be great, and leave us enough space for discussion.
Yes,
Thanks.
10 minutes it is. I can certainly do that. So the Water Framework Directive is under revision, and I'm just, I speed up a bit. One of the main things that is happening in the Water Framework Directive is a bit of a focus is now given to the problem of mixtures and the problem on how do we go beyond the substance by substance regulation. Here's one example, the PFAS compounds, where at least we're now starting to look at 24 together. It's just 24, but it's a good start, and we can actually extend that. The problem there is we do that for PFAS compounds, but we are ignoring a lot of the other groups of compounds where that should also be done, because we have groups of chemicals that should be regulated together. Okay, I was just asked to speed up, so I'm going to do that a bit, because also we've heard that before. The 8th Environmental Action Program has specific measures in place on biodiversity and ecosystems. And if we just look through the different indicators, we see that when it comes to human health, we actually are making a bit of progress. But when we're looking at the nitrates in groundwater as an indicator for environmental pollution, it's a flat line. When we're looking at birds, it's actually still decreasing. When we're looking at terrestrial protected areas, nothing is happening. Marine protected areas, nothing is happening. So basically what we're seeing here is we know what we should be doing, but we're just not moving forward fast enough there. The European Biodiversity Strategy was also mentioned earlier. So let me just very quickly mention one thing, that the European Parliament has been reacting to that. So the Biodiversity Strategy is a communication from the Commission. The Parliament has provided a resolution where they actually reacted to certain things. And here is one particular task to ECHA that was basically implemented, or that was basically a request from the Parliament. The Parliament regrets that the assessments of the impacts of chemicals on the environment and biodiversity tends to be underestimated and undervalued in the socioeconomic analysis during the authorization process. The Parliament calls on the Commission, in its role as a risk manager, to take greater consideration of the impacts of chemicals on the environment and biodiversity. So there's a clear task for ECHA to actually consider that within the instruments that are already in place, but this is a protection goal and protection value that we should perhaps take into consideration a bit more systematically. Okay, so now I'm already at the summary. So we have the recipient-oriented regulations. They are the counterparts of the substance-oriented regulations. But as mentioned before, I think the link between the two of them, that needs to be further developed. We really need to think about why are we having chemicals in our environment that shouldn't be there at those concentrations. I mean, we have demonstrated safe use, but obviously they are causing effects. So there's a disconnect there, and we need to act on that. The complexity of chemical pollution, the fact that we are seeing dozens or even hundreds of chemicals co-occurring in a river, in a plot of land, in air, etc., is really a problem. And this is really the challenge how do we do that? It's the challenge to achieve a good ecological status. It's causing what I think we also can call the silent pandemic. The silent pandemic is a terminology that comes from human toxicology, where people have been studying the neurotoxicity of industrial chemicals. And what you see is that we have a very good knowledge when we have incidents where individual ecosystems, in our case, are affected. When all the salmons are dying, we really see that. When a river is basically turning green, or when the oxygen concentration goes down, we can see that and we can act on that. So those local events, we have a good grasp on that, and we don't need to understand much more. We need to regulate that grasp better, but we have a good grip on it. But the broader the scope gets, we are having more and more subtle effects because we have a blanket of pollution everywhere. But that is only causing subtle effects, but they are happening on a continental scale. And that's the problem on how do we actually do that? So how do we act on that silent pandemic? So I think that's really the challenge. And that leads to sort of challenges that are sort of embedded in that. How do we handle the substitution of chemicals? How do we improve the single substance perspective that we are having at the moment? Because that leads to a substitution without any tangible improvement of the exposure in an ecosystem. How do we handle the need to prove causality under conditions of sometimes profound uncertainty? The example of diclofenac and the antioxidant in car tires, 6-PPD, that took years and really focused effort of a lot of people to find out what was causing that. And we can't do that for the thousands of chemicals that we need to work with. So what's our understanding of causality? When do we start to act, and when do we start to continue to explore an issue? I think that's something that we need to discuss and think about how can we improve that. So final slide, I think in order to move the needle, in order to actually go forward and improve things, we need to start to collaborate, perhaps a bit more systematic. We are used to do that already, but perhaps we need to also take into account all the new tools that we're having. So the main, say, disciplines there is certainly environmental chemistry, ecotoxicology, and ecology. And we need to make use of all those new tools, all the omics tools that we have, the machine learning and AI tools that we're getting, the EDA that I just showed you one example earlier on. So we have new tools at our disposal. We just need to sit down and think about how do we actually implement that into pragmatically useful approaches, but at the same time sufficiently protective when it comes to biodiversity protection. And with that, I'm done. Thank you for your attention.
And Romana, now you have a bit of a challenge. You also
Yes.
talk to us, and let's see if you're able to boil it down in like within a 5 minutes time window.
God.
That would be great.
Yeah, I try.
So we have an interactive seminar, I see.
The thing is, the pathway forward lies, as Thomas also said, we have to think across regulations, across different disciplines. We need, of course, the integration of the data, and we need a system understanding. And I try really to give some examples where PARC played a role, and I would also like to mention 3 other colleagues, Johan Axelmann and Peter van der Ohe. And there are actually at the moment a lot of opportunities. So as I said, we need a holistic approach for ecosystem level changes and the threats to biodiversity, and we can use NUMS and integrate NUMS. So we can use these data combined with, for example, high throughput DNA methods. But we need coordination. We need also validation to really support the regulation and the decision making. And one opportunity would be to establish next generation environmental risk assessment, which has, of course, the same level or even a better level of protection, including also the biodiversity. And within PARC, there are, so to say, different work packages, like the work package 5, dealing with the hazard assessment. So here are really new approach methods developed, and they are trying to fill knowledge gaps of purely characterized contaminants or new emerging hazards. And we can use and promote these innovative methods and tools to contribute to the integration of these techniques, of new technologies. And then we have, on the other hand, of course, we need to validate them. We need to integrate these methods into ecological validation. And this is done within work package 6 from PARC. And when we can validate these methods, we also, or we think that we are building confidence and we can implement it. So here's an example. Very different ongoing projects are developed, like high content screening in zebrafish embryos, or endocrine and neurotoxicity tests, or using also DEFNITs and so on. So you see a lot of things are ongoing in the work package 5, really to develop tests for replacements one-to-one, or you have a test battery, so to say. And there are a lot of different partners involved. So in silico methods are produced invertebrate models where we have at the moment no organisms in risk assessment in place. They are developed, and also to develop models like users on these based on the results there. So with those, I would like also to mention AI-assisted toxicity assessments, because there is a collaboration ongoing between the Yale University and the German Environment Agency, where they try to really look on all toxicity data across, so to say, the biodiversity, and to integrate all the data they have and to build up, so to say, persistency assessments, but not only persistency, but also the toxicity assessments. And there is one thing which is, I think, also really worth to mention, the BCF, for example, which is used under the biocides and also under REACH. The BCFs vary, and of course, the higher the accumulation is and the worse it is. And you see here, there are also different models developed, and perhaps in the future, we do not need any fish anymore or any Hyalella test for that. And this will be a publication within Science. It was submitted recently. And I think there are really promising methods and innovative methods out there. And we need a next generation risk assessment. First of all, we have to speed up the assessment. It's more ethic. We can focus on the most concerning substances. We are more efficient, and it should be one substance, one assessment. But the good thing is, if we are faster and when we know which substance is the most concerning one, or which groups, and we can establish also a framework where we take the ecology into account, we of course would better protect the biodiversity. So here there is a lot of things ongoing within BARC, but we need ecological validation. And I would like just to mention here one project. It's like that sediment cores are taken. Very often it's like that. You organize, you analyze the biodiversity, but you are not analyzing the chemicals which are out there. And with that approach, you go back in time and you look which chemicals have been in the sediment cores. You look on the species diversity, and you are training a model. You can integrate also temperature, pH, whatsoever, and you are modeling what could be happening in the future when you add a different chemical, or if you increase the temperature, because that's important. Biodiversity and climate change are interconnected. So this is a application. I don't want to get into details because I have no time, but there is, and I think this is also worth to mention. Normally, in a traditional way, we are performing laboratory tests like with DAFNITs or so on. And then we develop a threshold value. But the question is this the right way to go? And there are also discussions ongoing. Could we use an assessment factor which is field validated? And so this is one possibility and where a change could happen. And then I would like to mention also what is developed within PARC. All the methods, they should be, of course, integrated into the policy. So science to policy. And here is the work package 2.2. And you can have here also the link to the deliverable and contact also Matthias Herzler. So what is next generation risk assessment. It's further developed for human health, but there should be a next generation risk assessment also for the environment, also to better protect, so to say, the biodiversity. And with that, I would also like to mention there is a CTEC session, Biodiversity of Chemical Hazard and Risk Assessment. So please note the date and come and discuss it. We would be happy. And then last week, there was also issued, there is a special issue on that topic where we are looking for manuscripts in this area. How can we improve risk assessment, biodiversity protection with new innovative methods? And last but not least, what you see here is there are different projects ongoing, like Biodiversity Plus, Terrachem, CyberEC, Polynera, WildPosh, Norman, and so on. A lot of different projects also within the JRC, all dealing with biodiversity. And I think there is really the question, all these projects dealing on different topics. And there is, of course, interconnection. But the question is, do we need more dots, yeah? Or do we need to strengthen the connections? And we have one idea would be really to interconnect all these different approaches, all these different horizon projects, and to really, first of all, like integrate also the data, the opportunities, to engage the scientists, the regulators, proving of concepts which are there, and then to establish this further on. And with that, thank you. I had not that much time, but if you have any questions, we are a dedicated group working on that. Please contact us and we can give you more information. Thank you.
Good afternoon. Hello. Yes, it's Wim de Koen here. And actually, I want to thank you for the first historically, let's say, historically first scientific seminar that we're organizing. So I think it was a really excellent one with a lot of data, a lot of examples. Great to have some refreshing ideas and looking forward. First of all, I want to thank you as presenters, but also to give us ideas for future presentations. As you know, we're also involved in ECHA in PARC. And I think some of the slides that you showed towards the end for ongoing projects are ideal follow-up presentations. So your call will not be left in the void. We will come back to you and arrange follow-up seminars for sure. Now, just want to be, and the floor is open for others, of course, and I'll just kick off with the first question. So, you know, the political reality currently is quite different than 5 years ago. Yesterday, the Commission came with its Clean Deal that will go in parallel with the Green Deal. But you see a lot of, let's say, slight or too moderate to heavy shifts in policy focus. And while there's a lot of great research ongoing, this year, if something needs to change, this year will be the year that there may be potentially I'll call it a simplification of reach. So that's already maybe frightening for some of us if we hear that instead of adding more to reach, we will need to come up with some ways to simplify some of the aspects of reach. So we don't know yet what that fully means. We're having discussions with the Commission. But if I would ask you as researchers, knowing the problem of biodiversity with all its complexities, and I really appreciate all the slides because they're spot on. You talk about the substance-based approach and a recipient-based approach. If the Commission asks you, give me a simple tool now, today, to cover this problem, do you have something in mind? More research, AI, etc. I mean, we've all been in academia. That's usually the conclusion sentence, which we in the regulatory agency do not want to read, because we need to come up with a decision and a solution. So more research is not needed. What would be your way forward to deal with it now? I'm sorry to put you so on the spot, but I mean, we also want to hear some solutions from you. Thanks.
That's absolutely fine to put us on the spot. I guess that's why we're here. Can you hear me? Good. Okay. Yeah, of course, more data is always better, and more data allows you to also make better decisions. I think we have a couple of things that we can do before we are asking for more data. And the main, for me, one of the main things is to make better use of the existing data. I think that's really critical. And there are a couple of things in there. The one thing is, especially when we're looking at the political landscape and when we are looking at what's happening in the US, where they are now discussing to basically let go 60% of the employees at the US EPA, I think what we really need is a better organization, a better collection, and a better possibility to make use of the data that we are having. At the moment, they are scattered in too many different databases. It's a bit of a disaster almost that we have no database in Europe with toxicological, ecotoxicological data. We have the REACH registration dossiers, which is only covering the REACH data. We have the Open Food Tox database of the EFSA, but they don't talk to each other. We have no way to actually easily access the data and the scientific literature that are there. Whether we are going to use them in the end or not is another question. But right now it takes ages to actually collect those data all the time, although they are there. So both from an academic perspective, we need to become better at making our data easily available, machine readable, fair, so all those issues, but also from a regulatory perspective, we need to put those data together. We need to make them more available for risk assessment. And I think that already would move us a bit forward. And the one data that we are, or the one type of data that we're still struggling quite a bit with are exposure data. So ECHA should also be aware of the environmental monitoring data, and those data are changing over time, because when a REACH registration dossier is put in place and the PEC is calculated, by the way, which is something that is still not open for public scrutiny. So as an academic scientist, I usually don't get access to data that relate to production volumes and things like that. So the PEC calculation is a bit of a black box for us. But there has to be a mechanism in place that is going to be updated when we are gaining experience with those chemicals. So monitoring data, monitoring efforts, the AMPODAT database, for example, that Norman has put together, the monitoring data that the EEA is collecting in the water-based system, and all those things, that needs to come together better. So to make a long answer a bit shorter, make better use of the existing data, make them more systematically, make them more accessible. Let's use the stuff that we have for risk assessment.
Yes, I think it's really crucial to have the data available for the ones who need it. And this is one thing. But you were asking also about what could immediately happen. And the thing is, we cannot change science. Biodiversity decline is actually happening. So even if the European Commission changes or the political situation is changing, we will have that. We will have climate change, we have biodiversity decline. So that's happening, even if the political situation changes. So we have to deal with it. And I think there is an ongoing pressure from also now coming from the economic side, because biodiversity is an economic value, as I said, and it's within the next 10 years, it's one of the highest risks. So companies, they have invested, for example, for the reporting, because you were mentioning Ursula von der Leyen and the Clean Deal and the Green Deal, the reporting, also what's happening on biodiversity, pollution, and so on. Yeah. They have invested in that. So I think even if the pressure is at the moment not there, the industry has the pressure because they want to have money. They would like to earn. They would also avoid the risks, and they want to put their money into projects where the risk is actually avoided. And biodiversity is really on the top. So within other projects, we were discussing on CLP if there is a possibility, for example, for a new hazard class on biodiversity. But yes, it's even getting more complicated. But you could also have in the scope of REACH and the substance-oriented legislation, the scope biodiversity and ecology in, because at the moment it's like you only have it for the pesticides, but not for others, or an overarching. I mean, it's getting more complicated, but if you do not have the pressure, it something must happen. And I mean, I think the pressure it's coming from the economy also, but that's my opinion. And with the NUMS, I think there is a really huge opportunity because we can, it's getting faster. And of course, if you can screen more chemicals, it's getting also a little bit cheaper.
But there's one thing that we perhaps should also keep in mind, and the question is, are we seeing those biodiversity declines because we are lacking the tools and we're lacking the knowledge, or are we seeing those biodiversity declines because we are basically ignoring the tools and the data that we're having? It's a huge difference on whether we conclude the PNECs are not sufficiently protective for biodiversity, because then we need to think about how to better determine or to determine better PNECs, or whether we have the problems because we are exceeding the PNECs in the real world all the times. And that would mean we need better enforcement, we need better exposure assessments, and those kind of things. And that's two fundamentally different things. And I'm not aware of any situation where actually the PNEC has been demonstrated to be not sufficiently protective. I mean, the assessment factors seem to be doing a pretty good job. The problem is the enforcement, the implementation, and the conceptual gaps that the PNEC, the idea to have a PEC-PNEC ratio below 1, basically assumes that in your river or in your lake or in your soil, you only have one chemical, the chemical that you're interested in. The PNEC is the maximum concentration where we can demonstrate safe use for this particular chemical. But if there are 24 of other chemicals there, then we don't need rocket science to basically say, yeah, that might not really work. I mean, that's the same as if you're going into a pub and say, I can drink a beer, that's fine. Then you start to also drink a glass of wine, and a glass of champagne, and a glass of whiskey, and then in the end you are very surprised, oh shit, I'm drunk. And that's basically what we're doing here. So it's the conceptual problems, perhaps more than that we need more data, and maybe the way we are estimating PNECs isn't that bad.
Perhaps I jump in again, and for example, the exposure, the PEC calculations, and there is a study going on, as I mentioned, which is coming out in May, where actually the models getting more complicated, and when you compare the outcome with the measured values, it does not fit together. This is one thing. And the other thing is there is the study from Matthias Lies, where he actually investigated the effects on pesticides in rivers. And he came up with the idea that values actually which have been derived for no effect do not fit. We need a higher assessment factor, actually, if we take into account the ecology. So meaning that you would need for each pesticide, for example, a higher assessment factor. Not the one which was derived, but a higher one. And this is also published in 2 publications I have cited. So the question is really, did we validate the concept we implemented into regulation with real world data? And for the exposure, we can, if the report is coming out, then it's not really fitting. For the toxic threshold values, as Matthias Lies proposed, we would need a higher assessment factor, not the one which is now used. And this is one example. But the mixtures are actually not taking into account as such, despite we have all the regulations. We have biocides, we have pesticides, we have pharmaceuticals in the river. And there is no overarching cap, so to say. Where is enough? Where is it enough for the environment? Where is it enough for the biodiversity? And within the TerraChem project, there is also ongoing work to, perhaps there's a possibility to derive such an impact factor for biodiversity one could apply to see which chemical has which effect on biodiversity. So there is research ongoing.
Well, everyone seems extremely happy and yeah, with your presentation here. Thank you so much, Romana and Thomas, and thanks everyone who made it to the seminar. And yes, well, and we will very soon send a survey to all the participants and hopefully get useful feedback to be shared with our lecturers. And with this, I would like to close this first ECHA Science Seminar. And yes, see you soon again. Thank you. Bye.